Hello and welcome, one and all, to Antiviral Roller Skating. I know I'm not on skates, but this show is not about me. I want you to take your mind back to the summer of 2020. Bit of an awkward time, wasn't it? But some people picked up roller skating. Some even picked up the high-impact sport of roller derby, could you imagine? But I bet they didn't wonder what a skating virus might look like. So, without further ado, let's get this show on the roll. Once upon a time, there was a virus. You might remember them. SARS-CoV-2, aka the bane of our lives since 2020. A nasty piece of work, am I right? Anyway, I've gathered you all here today to tell you a totally rad story, how scientists worked hard to design drugs against this troublesome foe. So let's give it up for our drug heroes, Pax and Rem. Now we've met our main characters, let's see what they are fighting for, the cells in your airways. Imagine someone open-mouthed sneezes on your daily commute, and try as you might to close your nostrils, there's just no hope. Those sneeze droplets are travelling at your face at 100 miles an hour, and you breathe them in. The virus finds its way into the cells of your airways, and so the mischief begins. Now I'm not sure how many of you have ever seen the inside of your cells, so let's help you imagine, shall we? First, we have the cell membrane, which controls all transport in and out of the cell, a bit like a gated fence around a property. If anything wants to get into this property, like a virus, it better have the right key. As you'll see, the cell membrane has different projections on the outside, known as receptors. Think of these as the padlocks on the fence. The inner space of the property, or the cell, is referred to as the cytoplasm. Then, we have the nucleus, the control center of the cells, containing your DNA, which contains instructions to make every protein your body needs. Want to make proteins? Then you better have ribosomes. These clever components convert your genetic instructions, DNA, into proteins using a messenger molecule called RNA. And finally, we have the endoplasmic reticulum. You can call it ER if you're nasty. Now the ER is where many of the ribosomes will be, producing lots of proteins. If anything wants to escape the cell, including viruses, it usually must travel through the convoluted corridors of the ER, getting packaged up and transported to the cell membrane for release. So, what happens when a virus gets in? Let's find out.
Well. What do we have here? Looks like a perfect place to set up shop and make clones of myself. Now, how do I get in?
The virus must enter via a receptor. A receptor is a protein or a sugar bound to the outside of the cell. Think of these like a padlock. The virus needs the correct key, which are the spiky crown-like projections that decorate its surface. It uses these spikes to open the padlocks. The virus unlocking the receptor will trigger a change in the shape of both the virus and the cell membrane, allowing the virus to enter the cell. Once the virus is inside the cell, it releases its genetic material, also known as the viral RNA genome. Once the RNA genome is in the cytoplasm, it can hijack the cell's ribosomes to produce its own proteins. How rude! This process is called translation. Think of it as changing the language of RNA into the language of proteins. For coronaviruses, everything they need to replicate themselves is produced as one long protein. This long protein contains an enzyme, a pair of molecular scissors if you will, that can release itself from the long protein chain and then start releasing other proteins as well. Now, the virus has all the proteins it needs to reproduce itself. Some of these proteins assemble together to make a molecular printer. And this printer, also known as the replication complex, is responsible for making many copies of the viral genome. Once the printer has copied enough genomes to make many proteins, these proteins assemble into a new generation of viruses. These viruses are not always exact copies of the initial virus, but may acquire small changes to their genome. These new viruses will package up a single genome each and exit the cell to go searching for new cells to infect. So yeah, that's how a virus causes chaos in our cells, which makes us feel under the weather. So what can our superhero drugs do to stop it? First up, we have Paxlovid, or Pax for short. This drug is designed to stop the virus's molecular scissors from working, causing the viral proteins to get stuck in the long protein chain. The virus is then unable to assemble the molecular printer to produce more genomes, stopping the virus in its tracks. Disarmed, the infection is stifled, preventing the virus from spreading to other cells. Some patients cannot take Paxlovid as it interferes with their other medications. So, there are other drugs, such as remdesivir, or Rem for short. This drug acts at a different stage of the virus life cycle. This time, the molecular scissors can still function and the molecular printer is assembled. But then, Rem comes along and jams the printer, causing it to misprint genomes. This leaves the virus unable to reproduce its genome accurately, leading to faulty viral RNA. As a result, fewer new viruses are made, eventually ending the infection, and the cells survive. So, how did we create these drugs? Well, scientists have been investigating these viral proteins for decades to gain a greater understanding of how they work on a fundamental level. Understanding exactly how the proteins interact with each other allows chemists and other drug developers to design antiviral drugs more effectively. This fundamental research underpins many developments in modern medicine and requires sustained effort and funding. Another side to this story is the many wonderful humans who volunteered to take part in clinical trials to test these drugs' effectiveness against SARS-CoV-2 throughout the pandemic. In signing up for clinical trials, they help doctors and scientists to characterize exactly how these drugs work in people and which communities might benefit the most from targeted treatment. One downside to many clinical trials is that the diversity of participants is often skewed, with participation rates lowest amongst historically marginalized groups. This can often be due to a long history of medical mistreatment, leading to generational mistrust of experimental medicine. So much more can be done to increase equitable access to life-saving antiviral drugs, which will be crucial to our response to future pandemics. This work starts with scientists and policymakers unpacking the complex dynamics underpinning medical racism, health disparities, and systemic injustices within our health services. The joy of scientific discovery is a joy that needs to be shared equitably, as well as the benefits these discoveries bring to the advancement of human health. Can you imagine how many years of research went into the medicines in your bathroom cabinet? It all started with asking, what if we could treat this? We now need to be asking, how can we get this medicine to everyone that needs it? Thank you for rolling with us on this skating adventure through the trials and tribulations of antiviral discovery. As we say in roller derby, the pack is here, the pack is all, meaning stay together and we can fight this.